been diagnosed with cancer so scary? Cancer is scary because it's so familiar. It's not just that we all know someone who's had cancer, but it's that we all know someone who has died of cancer. So how are normal cells so different from cancer cells? Normal cells have all the right checks and balances that allows them to have the right size, maintain the right shape, and as a result, that prevents them from going out of control. On the other hand, cancer cells lack all these checks and balances, and that enables them to grow out of control. Most of these checks and balances are roles that are played by proteins. And mostly in cancer, these proteins are either overexpressed or not expressed at all. So understanding the big roles that these proteins play in cancer would help us to design better drugs that can treat cancer. There are several types of cancer, but lung cancer is a leading cause of cancer-related deaths worldwide. And about 35% of the most common form of lung cancer has a mutation in a protein that is known as liver kinase B1, or LKB1. So you can think of LKB1 as a protein that is expressed in all cells, and it maintains cell energy levels, cell growth, and it's able to function as a tumor suppressor. So you can think of LKB1 as an energy sensor. So in the cell, LKB1 is able to regulate processes that produce energy, as well as processes that consume energy in order to maintain a balance. So think of it this way. Cells with LKB1 are like athletes in a marathon with checkpoints. So these checkpoints are there to prevent casualties and enable the athletes have a successful race. So they know when to stop, they know when to grab a drink, and they even know when they arrive or when they get to the finish line. On the other hand, you can think of cells without LKB1 as athletes in a marathon with no checkpoints. So the impression here is just to finish the race. So all they do is just run without any proper way of regulating their energy levels. So ideally, one would think that without being properly able to maintain your energy levels, this should lead to casualties. But this is not the case in cells. So you think that cells that are not able to properly regulate their energy should easily die. But that's not the case. It's actually been observed that cells without LKB1 are more aggressive, and they're able to spread from the lungs to other vital organs. So the question now is, what is the support system? What else is helping these cells to be able to grow out of control and spread to other vital organs? So how do we go about this in the lab? We culture cells that only have one variable, which is LKB1. So we have cells that have LKB1 present and cells that do not have LKB1. And then we look for all the molecules possible that could be contributing to the role of LKB1 as a tumor suppressor. So in this case, we extract proteins as well as metabolites, or all the small molecules that are in the cell. And then we use mass spectrometry as a tool to help us identify the molecules. So you can think of mass spectrometry this way. So just like your mailman is able to sort and identify your mail by your zip code, mass spec is able to sort molecules by their exact masses. And then with the help of software tools, we are able to identify the individual molecules. So being it proteins, lipids, or sugars that would be in these cells. And then we use statistical tools to look at the levels of expression of these molecules. So if they have a specific protein present in both cells, are they the same? Or is one higher when LKB1 is present versus when LKB1 is absent? And if the protein is high, what big role is it playing in the cell? So we use pathway analysis to kind of understand what big processes these proteins or small molecules might be playing in the cell. So in our results so far, we see a lot of processes that are being controlled by our proteins and metabolites that we find. And one of such pathways that is very prominent when LKB1 is absent is the Mtor pathway. The Mtor pathway is simply the protein synthesis pathway. So when LKB1 is absent, these cells continuously turn on this pathway to keep making proteins. And this is intuitive because as humans, we all need proteins to grow. And the cells have found a way to keep making proteins to help them survive even without a proper way of regulating their energy levels. Another pathway that is prominent when LKB1 is absent is the KEAP1 NRF2 pathway. So this pathway is simply the stress pathway and is affected by activity as well as our diet. So in cells without LKB1, we see a lot of stress molecules or reactive oxygen species, as well as a lot of antioxidant proteins, which helps to scavenge these reactive oxygen species from the cells. So now we know what these cells are making. So we have targets. We know what is present when LKB1 is not in these cells. So in our ongoing experiments, we are looking to validate these targets in resected tumor samples from lung cancer patients, as well as doing other biochemical assays to understand exactly how these targets or how these molecules might be playing a role in lung cancer. There are currently no FDA-approved drugs for LKB1 mutated lung cancers. And LKB1 mutations are also found in other cancers such as cervical cancer, breast cancer, as well as skin cancer. So our hope is that the discoveries made from the study would contribute to the pool of knowledge on the understanding of the roles of LKB1 to help create more effective drugs to target cancer and to help create a cancer-free world. Thank you.